Hello guys, I welcome you all to the NEET Botany Sucharita. Today in this video session, I'm just going to make every NEET aspirant of course 2026 class 11 and class 12 about the light reaction. Especially I'm just going to discuss part of the light reaction which is electron transport chain. And simple form I'm just going to call it as ETC. Usually the ETC constitute both non-cyclic and cyclic pathway. Today I'm just going to take the non-cyclic pathway, right? So you need to understand in the membranous structures there are photosynthetic pigments, and these photosynthetic pigments, especially in the higher plants, are of two types. One is photosystem 2, another one is photosystem 1. And make sure that try to understand. Can you see here? I have taken a small thylakoid structure. I have taken here thylakoid. I'm just going to cut into two pieces, or just I'm going to take out the small portion of the thylakoid. In the small portion of the thylakoid, assume that there are photosynthetic pigments. Yes, PS, photosynthetic. Yes, photosynthetic pigments. Yes, there is presence of photosynthetic pigments are there. And these photosynthetic pigments, as I have discussed in the previous class, so shall I just say, are they scattered like that? No, they are not scattered. So how they were? So they were just grouped. Yes, they were grouped. They were grouped, and then the group of photosynthetic pigments, today we are going to call them as photosystems. Yes, what we are going to call them as the group of photosynthetic pigments which involve in photosynthetic action, especially in the higher plants, are going to have two photosystems. What do you say? What do you say? Please tell me. In the higher plants, so there is presence of photosystem 2 and there is presence of photosystem 1, and this two types are just on the basis of order of discovery, the sequence of discovery. Please don't forget. So this photosystem 2 and photosystem 1. These are the two types of photosystems present in case of higher plants. Guys, if I'm just talking about the photosystems, yes, any photosystem, let me write here for you, any photosystem, be it 2, be it 1. The photosystem, be it 1 or be it 2. So it constitutes two regions. Yes, it constitutes how many regions? Two regions. Yes, they constitute the two regions. Don't forget. One is the reaction center. The reaction center. I'm just going to make the video on this in upcoming sessions. Another one is light harvesting complex. LHC. What do you call this complex as? Light harvesting complex. Sorry, what is this? Light harvesting complex. So each photosystem constitute two regions. One is reaction center, the other one is light harvesting complex. Please don't forget about it. Now I'll tell you, the reaction center constitute only one photosynthetic pigment, which is nothing but chlorophyll A molecule, and this is going to be in how many molecules? There is presence of single molecule of chlorophyll A is there. So photosystem 1 and photosystem 2, they constitute the two common regions. One is reaction center, another one is light harvesting complex. The reaction center constitute the chlorophyll A of single molecule, but the light harvesting complex constitute chlorophyll B, carotene and xanthophyll, and the number are going to vary from hundreds to thousands. So let's say, repeat this point, the light harvesting complex constitute other than the chlorophyll A. Other than the chlorophyll A, we are having the chlorophyll B, carotene, xanthophyll, and the number is going to be not single molecule. The number is going to be hundreds and thousands. Is this clear? How many types of photosystem? See, can you just see? I have taken the thylakoid. I just took a small portion, and I'm saying the photosynthetic pigments are not scattered. They're going to be in grouped, and these group of photosynthetic pigments are called as photosystem. And in the higher plant there is presence of two photosystems. One is PS2, the other one is PS1. The PS1 and PS2, or PS2 and PS1, there comes two regions. One is a reaction center, you can see it very clearly. One is the reaction center, the other region is light harvesting complex, and this reaction center constitutes chlorophyll A of single molecule, and the light harvesting complex constitute chlorophyll B, carotene, xanthophyll in hundreds and thousands. I hope you understood the brief discussion about the photosystem. Now I'll just show you the arrangement here. Can you see? I have taken the small portion of the thylakoid membrane. Yes, I have taken a small portion of the thylakoid membrane. Can you see in the thylakoid membrane there is presence of photosynthetic pigments which are not scattered. They are grouped as PS2 and grouped as PS1, right? And you know that the PS2 and PS1 constitute two regions. One is reaction center, the other one is light harvesting complex. So I have taken the PS2 here and PS1 here, right? So now you can see between the PS2 and PS1 there is presence of electron transport carriers are there. Electron carriers are there. One is plastoquinone, the other one is cytochrome BF complex, the other one is plastocyanin. I repeat, between the PS2 and PS1 there is presence of plastoquinone, cytochrome BF complex. I just wanted to tell you a very important three things. This is an electron, this is an electron carrier, this is an electron carrier. How many electron carriers are there? Plastoquinone, cytochrome BF complex and plastocyanin. But among these three electron carriers, the one which is also have ability to pump the proton, also have ability to transport the hydrogen, is nothing but plastoquinone. That's why people are calling this one as hydrogen carrier, or it is also called as proton carrier, proton pump. Yes, did you understand what I am trying to say? Now let us start here. What happens is the very first step, yes, the very first step of the non-cyclic ETS is excitation. This should be very clear. The very first step of the electron transport system is excitation of, try to understand, I'm just teaching you in a very slow pace. Excitation of PS2. Yes, excitation of the PS2 will occur when it excites by just absorbing, right, 680 nanometers of sunlight wavelength trapped by the chlorophyll A containing in the reaction center. Yes, the reaction center of the PS2 is having the chlorophyll A which get excites by absorbing 680 nanometers. Is this clear what I'm trying to say? What happens? What is the first step? It's not water splitting, madam and sir. It is all about the excitation. Excitation of the photosystem 2 occurs at the 680 nanometer by the chlorophyll A present in the reaction center. I repeat this point. The reaction center, which is made up of chlorophyll A, excites at 680 nanometers of the PS2, right? So now what happens in the second step is, after excitation, the reaction center containing the chlorophyll A is going to show extrusion of electrons. Yes, it is going to extrude the electrons. When it gets extruded the electrons, try to understand. So on losing the electrons, it's going to move from ground state to excited state. Try to understand. Right? So the chlorophyll A, after absorption of the 680 nanometer, it started showing extrusion of electrons. After extrusion of electrons, it is going to release electrons from the ground state to excited state, which means it is losing the electrons. Now after losing the electrons, the chlorophyll A molecule is going to act like a strong oxidant. Then it is going to be acquiring the positive charge. Yes, it is going to acquire the positive charge, and then I'm just going to call it as P680 plus. Yes, now it is a strong oxidant. I just show you in a diagrammatic way. Just listen carefully. I'm going to represent here what I have written. Now let's start here. Here comes sunlight. The PS2 is going to absorb the 680 nanometer. How much nanometer is going to absorbed by the PS2? Of 680 nanometers. Please don't forget. Please don't forget. It's going to absorb the 680 nanometer. Now the PS2, which contains one of the region called reaction center containing the chlorophyll A, is going to extrude the electrons from ground state to excited state. What do you say? Yes, excited state. Now there's someone should be there. I'll tell you. The reaction center losing electrons. And now this reaction center containing the chlorophyll A is going to acquire the positive charge. And I'm just going to call this one as P680 plus. Right? Now it is going to become a strong oxidant. So now you can see here very clearly. I'm just going to tell you specially for you guys. This PS2, especially the PS2, not PS1, phasing the lumen, phasing, this one is considered to be as thylakoid membrane, because this is the thylakoid membrane. I'm just going to cut here. After cutting here, this portion, I'm just going to call this one as thylakoid lumen. We are going to call this one as thylakoid lumen. So try to understand, this one is considered to be as thylakoid lumen. This thylakoid lumen is considered to be as water splitting complex. What are you going to call this one as? Water splitting complex, right? You just try to understand. So now let's start. The sunlight is going to be absorbed. The sunlight with 680 nanometers is going to be absorbed by the photosystem 2. This reaction center containing the chlorophyll A of the PS2 is going to excite by absorbing how many nanometers? 680 nanometers, and extrudes electron from ground state to excited state. After extrusion, it is going to acquire the positive charge. Now this acts like a strong oxidant. The strong oxidant is going to involve in oxidation step. Yes, it is going to involve in oxidation. It's going to split the water. Yes, the third step is Water splitting, right? So let us call it as water is undergoing oxidation. Water is undergoing what? Oxidation. And on oxidation, the water has to release electrons, protons and oxygen. Can you try to please recollect the points? The chlorophyll A, after losing electrons from ground state to excited, the chlorophyll A in the reaction center is going to acquire the positive charge and acts like a strong oxidant. Right after acquiring positive charge, it acts like a P680 plus, which is going to oxidize the water. Let's say this step as water splitting or oxidation. In turn, releases electrons, protons and oxygen. Yes, now you need to understand here. Can you just see? So now the water is undergoing oxidation. The water is undergoing oxidation to release electrons, protons and oxygen. Now people are asking you, my dear, the oxygen is not released somewhere else. The oxygen is going to be released inside the lumen as a byproduct of the photosynthesis. Is this okay what I'm trying to say? Now you got the electrons and protons in the lumen. Now these electrons need to pick by this PS2. And now immediately the electrons which are released, I'll just show you, don't worry. The electrons which are released, yes, is going to accept, electrons are going to accept by, this is very important point, the first electron acceptor of PS2 is not Sucharita. It is nothing but pheophytin. Repeat this point. The electrons which is extruded by the PS2 is going to be accepted by the very important first electron acceptor, which is nothing but pheophytin. So if people ask you this point, the first electron acceptor of the PS2 is dash pheophytin. Now the electrons are from where to where? From the reaction center to the pheophytin, which is present inside the PS2. Now the electrons were given to the electron carrier, proton carrier, hydrogen carrier called plastoquinone. Now this plastoquinone is going to carry that electron from the PS2. Not only that, it's going to pick how many protons? 2 protons from the stroma or matrix. Now try to understand. I'm just going to repeat this point. The plastoquinone, which is a hydrogen carrier, is going to pick the electrons from the PS2. After taking the electrons from the PS2, it also picks the protons or 1 hydrogen from matrix. Right. Now with the plastoquinone, there is not only presence of electrons, also there is presence of hydrogen. It tries to give to the cytochrome BF complex. Yes, but the cytochrome BF complex is accepting only the electrons but not the protons. Hence, this plastoquinone is going to pump those 2 protons or those 1 hydrogen from matrix into the lumen. Don't forget, into the lumen. So try to understand, the plastoquinone is a hydrogen carrier which picks electrons from the PS2 but also picks protons from the matrix, and both electrons and protons, when it tries to give to the cytochrome BF complex, it just takes only electrons, hence rejects protons. That's why the plastoquinone is going to pump those protons from matrix into the lumen. Please don't forget. Now the cytochrome BF complex, which carries the electrons, will have to give to the next electron carrier. It's nothing but plastocyanin, which is the last electron acceptor. Now the plastocyanin has to give electrons to the PS1. But the PS1 should be an oxidant. How it acts like an oxidant? Only it acts like an oxidant when it absorbs 700 nanometers of wavelength of light. Yes, 700 nanometers is going to excite the chlorophyll A present in the reaction center of the PS1. Same thing repeats. It's going to extrude the electrons from the ground state to excited state. Now it is going to acquire the positive charge. I am going to call this one as P700 plus. Now it is going to act like a strong oxidant. As I said, the oxidant can oxidize. Now this oxidant is ready to pick the electrons from the plastocyanin after excitation, and then it is going to give the electrons to the first electron acceptor, which is nothing but not a pheophytin. It's nothing but iron sulfur complex. Yes, if people ask you, the first electron acceptor of the PS1 is not a pheophytin, it should be electron acceptor, that is FES, iron sulfur complex. Now the electrons are with whom? The FES. The FES is carrying the electrons, is going to give to them another electron carrier, where I'm just going to call this one as ferredoxin, which is present facing towards, not to the lumen, facing towards the matrix. So please don't forget. To the matrix, to the ferredoxin, another enzymatic complex which is associated here is NADP reductase. Please don't forget this point. I'm just going to write. So what is there? Which enzyme is there? NADP reductase is attached with the ferredoxin. Yes. Now the electrons with the FES is given to the ferredoxin. Now the ferredoxin is going to give this electrons to the NADP plus. How many electrons it is going to give? That is going to be showed in next session. The NADP plus is going to accept the electrons from the PS1 and also accept the protons right from the matrix. 2 protons are going to be accepted from the stroma or matrix. So NADP plus is going to accept the electrons from the PS1 and 2 protons from the matrix, and after gaining it's going to become NADPH plus H plus. What you say? The electrons are contributed by the PS2 and transport to the PS1, and it is utilized for the synthesis of NADPH but not returned back to the PS2. That's how I'm just going to call this ETS as non-cyclic ETS. So please try to understand the non-cyclic ETS is requiring the 2 photosystems, both PS2 and PS1. The electron carrier is the water, and the water, which is electron contributor, is not attached with the PS2, it is attached with the PS1 facing towards the lumen. But here you can see ferredoxin is attached to the PS1, not to the PS2, by facing towards the matrix. Now the electrons which are contributed from PS2 is transferred through all these 3 electron carriers right towards the PS1, never given back here in the non-cyclic ETS. Hence it is retain these electrons right for synthesis of NADPH, one of the energy rich molecule contributed by the light reaction. If you understand this concept, please don't forget to subscribe the channel, because your hitting the likes and subscribing this channel will help you a lot of people who are just falling back beyond or scary about the plant physiology. So always Sucharita.